Chemical reactions usually occur in a series of reactions called a metabolic pathway. A pathway has a specific starting reactant and ending product. In this example, the starting reactant is molecule A, and the final product is molecule D. The pathway takes 3 different reactions to go from A to D. Each reaction is catalyzed by an enzyme. Without the enzyme, the reaction would not occur fast enough for your body. If one of the enzymes in the pathway isn't available, then the pathway will stop working and we won't be able to make product D. Enzymes are a type of protein that catalyze reactions. This means that they make reactions go faster. Enzymes do not affect the total free energy of the reaction. However, they can make reactions go faster by lowering the energy of activation. The energy of activation is a barrier that prevents a reaction from starting. The enzyme can reduce the barrier and make it easier to start a reaction, making it occur much quicker. In this example, the blue line indicates the reaction without an enzyme, while the yellow line is the reaction with an enzyme. Notice that for both lines, the reaction starts and ends in the same place. The difference is the energy of activation, which is much lower for the yellow line. So the reaction can proceed fast with help of the enzyme. Enzymes work because they have a unique 3D shape. A substrate, which is the reactant in the chemical reaction, can fit into the active site. Each enzyme is very specific to only certain substrates. If a substrate is slightly different in shape, it will not fit into the active site. When a substrate binds to the active site changes shape very slightly. This is called the Induced Fit Model. This change in shape holds on tight to the substrate to get the best fit between the substrate and the active site. Once the reaction is complete, the enzyme releases the substrate, which is now the product of the reaction. The active site returns to its original state, and now the enzyme can be used to do that reaction again. Enzymes work very quickly. Catalase can break down hydrogen peroxide 600,000 times per second. The amount of substrate available can alter how quickly a reaction proceeds. When not much substrate is present, the reaction goes slowly. The y-axis here shows the speed of the reaction. When the substrate concentration is low, the reaction speed is low. As more substrate is added, the reaction begins to speed up. Eventually, the reaction will not be able to go any faster. Once there is more substrate than enzymes, the reaction is saturated and it can't go any faster. Adding more substrate doesn't increase the speed anymore because there aren't enough enzymes for the extra substrate. Temperature and pH also affect the speed of enzymatic reactions. The black line on the temperature graph shows the speed of the reaction for a human enzyme. At 0 degrees Celsius, the reaction is stopped. As the temperature increases to about 37 degrees Celsius, the speed of the reaction increases. But when the temperature increases again to 45, the speed of the reaction quickly drops back down to 0. The temperature in the body got too hot and the enzyme was denatured. This means it changed its 3D shape and it cannot work anymore. The red line on the temperature graph shows an enzyme that lives in a heat-resistant bacteria. This also follows the same pattern. It has an optimal temperature that it functions the best at, about 77 degrees Celsius. When the temperature is lower or higher, the reaction does not go as quickly as at the optimal temperature. The same pattern can be seen for pH. Pepsin is an enzyme in your stomach. It works best at low acidic pH levels. The enzyme trypsin in your small intestine works best at higher, slightly basic pH level. When the pepsin from your stomach moves into your intestine with your food, it is denatured. Not all enzymes are needed by your cells all the time. Enzymes can be activated or inhibited depending on which reactions need to be catalyzed. An inhibitor turns an enzyme off and stops it from working. The inhibitor will bind to the enzyme, often in the active site, and change the enzyme's shape. When the shape is changed, it can't function anymore. NSAID pain relievers work because they inhibit enzymes. When they block the enzymes from making prostaglandins, it helps prevent pain and swelling in your body. Feedback inhibition is how your body regulates its metabolic pathways. Here's the same pathway that starts with reactant A and ends with product D. Product D inhibits enzyme 1. So when the body has made enough of product D, it inhibits enzyme 1, which shut down the pathway and stops making it. Eventually, when the body uses up all of product D, the inhibition stops and the pathway can resume making product D. Cofactors and coenzymes are small molecules that bind to enzymes and help them work properly. A lot of the minerals and vitamins in your diet are important because they assist enzymes in your body.